My name is Shazlyn binti Ismail.
Hi, my name is Fasihatul Husna binti Faizal.
Hi, my name is Sharifah Khalisah binti Said Alawi.
Have you ever wondered why does hot pepper is so spicy? The answer is hot pepper contains a substance called capsaicin. When the substance comes in contact with pain receptor in your mouth, it will produce a burning sensation. But did you know capsaicin have a lot of benefits like increasing metabolism by burning fats and reducing insulin spikes in diabetes? So how to extract this capsaicin? In this video, we will explain everything about how to extract capsaicin by SOXHLET extraction.
History of SOXHLET extraction. Franz Ritter von SOXHLET, this German agricultural chemist, was the first to introduce its SOXHLET laboratory extractor in 1879, which deals in the determination of meat fat. The method was a significant advance in the isolation of solid from difficult to extract solid sample. Since then, this extraction technique is widely used for fats and oil, as well as the other mostly non-polar solid from solid and semi-solid matters, because its efficiency of extraction is greater than that of other manual techniques, especially at the time. It is also gained broad acceptance. Why solid extraction are important? It is required where the desired compound has only a limited solubility in a solvent and the impurity is insoluble in that solvent. If the desired compound has a high solubility in a solvent, then a simple filtration can be used to separate the compound from the insoluble substance. Chili peppers are generally known as ripened fruits of various species of genus Capsicum. Their hot sensitivities is due to capsaicinoids as a major group of organic compounds which is closely related to the family of alkaloids and are known to be biosynthesized and accumulated in the placenta of capsicum fruits. Capsaicinoids are soluble in moderate polar organic solvents like chloroform, acetone, ethyl acetate, methylene chloride, methanol, ethanol, aceton nitrile, and others. Why solid extraction was used in this experiment? This is because SOXHLET are more appropriate compared to another methods of extractions. Solid extractions allow use a large number of organic solvents, including ethanol, which may be accepted for pharmaceutical purpose. The fundamental nature of solvent extraction was that it enables a compound or mixture of compound to be extracted at a temperature substantially below that the boiling point of the individual constituent.
How does SOXHLET extraction work? First, we turn on the heat, then the metal plates gets heated. The round bottom flask which contains the solvent will start boiling. The vapor from the flask will travel to the condenser via the distillation tube. The condenser condenses the vapor of solvent. Then, those condensed vapors will fall down to timber and cause the sample wet. The sample gets wet with the solvent component. The solvent mixture starts filling timber and siphon. A point reached where the siphon starts overflowing under the influence of gravity. Since the siphon directly connects flask, the overflow liquid falls back to flask. This marks the first cycle of SOXHLET extraction. For each cycle, we don't have to change the solvent. After a few cycles, we are left with the mixture of solvent and the components from the sample which are soluble in the solvent.
The equipments that will be needed for this experiment are round bottom flask, heating mantle, rubber tube, rotary evaporator, and timber. We are also required materials such as 14 grams of dried chili pepper as our sample, 250 ml of ethanol, and boiling stone, while apparatus for the extractions are SOXHLET extractor and condenser. The type of sample is plant tissue since we will be using dried chili pepper, so our sample is dried chili pepper.
So, the first step is weigh 14 grams of dried chili pepper. It is critical that the pepper be as dry as possible, or else you will extract a lot of water as well as capsaicinoid. Excess water will make drying the extract take much longer. Pepper must also be ground as finely as possible. If you buy fresh pepper, simply cut the pepper and dry it thoroughly in an oven before using them in the extraction. Then, the second step is put the sample which is the dried chili pepper into the timber. The dried chili pepper should fill approximately 1/3 of the timber. The timber is made of cellulose, which is the same material used to make filter paper. So, this is simply a piece of filter paper molded to fit the well in the SOXHLET extractor. The thimble also ensures that the rapid motion of the solvent does not transport any solid material to the steel pot. After that, step 3 is prepare the extraction solvent by filling the 250 ml of ethanol into the round bottom flask. The solvent you choose must be anhydrous, which means it must contain little to no water, which include water in the solvent that will reduce the solubility of the capsaicin, but also make the purifying your extract more difficult. After that, step 4 is close the top of the sample, which is the dried chili pepper with the cotton wool inside the thimble. Normally, a cotton wool is placed on top of the sample in the thimble to secure its position within the thimble, as the sample may float out of the thimble. Then, step 5. Add some boiling stone into a round bottom flask that filled with 250 ml of ethanol. A boiling stone is a small, unevenly shaped substance that is added to liquid to facilitate boiling process by making them boil more slowly or calmly.
Step 6 is to insert thimble into extraction apparatus. The thimble, it will be clamped into the extractor after being filled with the sample. Step 7 is to assemble the extraction body. Assemble the round bottom flask, SOXHLET extractor and condenser on the second stand above the heating unit. Step 8 is to switch on the heating mantle. Make sure to raise the laboratory stand until the round bottom flask is surrounded by the mantle and carefully boil the ethanol. Observe it and adjust the settings in case the boiling rate is too fast or too slow. Next step is to open the tap water. Rubber tube is attached to the two ends of the condenser. Then turn on the tap water and make sure that the water enters the spiral tube and leave through the vertical tube. This will create a counter-current cooling, so make sure that the apparatus is stable and the water flow will be endless during the extraction process. Step 10 is to wait until a few cycles. The cycle of extractions, where the solvent will fill the main chamber and dissolve the desired compound from the dried chili paper, is repeated few times because each time the extraction is repeated, it will cause more of the dried chili paper to dissolve. This will leave the insoluble impurities in the thimble and how our desired compounds will be removed from the dried chili paper. This cycle is repeated for at least 30 to 40 minutes.
After a few cycles, we will eventually come to the end of the extraction. So, how do we know that it's the end of the extraction process? It is actually indicated by when the extraction solvent will run as a clear solvent in the SOXHLET extractor. It is no longer a colored solvent as how it initially was. After the extraction process has completed, what we would do is we would collect the final solvent in the round bottom flask, take it out and let it cool for about a few minutes. Next, put the round bottom flask containing the ethanol on a rotary evaporator and switch it on. What does the rotary evaporator do? It is used to remove the solvent from the sample through evaporation, and the rotary evaporator would allow a quicker evaporation compared to normal evaporation process. After about 2 minutes, switch off the rotary evaporator by switching off its aspirator. Then carefully take the round bottom flask from the rotary evaporator and observe the result. Now let's look into what we might observe after the extraction. If you want to solidify it more, you might want to put the sample in ice packs for a few minutes. But this is basically how the result should look like after the extraction. A yielded oily red extract can be seen in the flask, and that is actually our capsaicinoids. It is red colored due to the presence of beta-carotene. This extracted capsaicinoids is very strong. It can burn skins, and that burn can last from up to 1 day, so it's very essential to use gloves when handling capsaicinoids.